So, students, today's topic is embryonic development up to blastocyst formation, and this topic has been taken from Unit Reproduction, which is in our syllabus for class 12th in zoology subject. Before moving ahead, you should have a little bit information what embryonic development is. Embryonic development, by name, it indicates the development of embryo. Firstly, when there occurs the fusion of haploid male gamete, that is sperm, and haploid female gamete, that is egg, inside the fallopian tube of mother, a diploid zygote is formed. So this diploid zygote undergoes successive mitotic divisions, not meiotic but mitotic, because it have to now maintain the number of chromosomes in diploid nature, that is in 2n number. So the zygote undergoes successive mitotic divisions in order to form firstly embryo, and then fourths, and then lastly the complete full term fourths. So this all is embryonic development, but today we have to read embryonic development up to blastocyst formation. So embryonic development, it undergoes by a process, by a phenomena which is known as cleavage. So first of all, we should know what cleavage is. So cleavage is the process where a single celled zygote is diploid in nature, because there occurs the fusion of haploid sperm and haploid this egg or secondary oocyte. Now diploid zygote is formed, but this diploid zygote is a single cell. So when it undergoes repeated mitotic divisions in order to give rise to a multicellular structure which is known as blastula or blastocyst, and this phenomena or this process is known as cleavage. So students, cleavage, it is the process where a single celled diploid zygote undergoes mitotic divisions in order to give rise to a multicellular structure which is known as blastula or blastocyst. So this is the cleavage, and now we should now know how this cleavage, how this process starts and how this proceeds on. So here is a pictograph of this. This is the ovary of mother, where eggs or secondary oocytes are formed. Here it is mature follicle, where at the ovulation stage, under the influence of luteinizing hormone, its wall gets ruptured, which I have already taught you in this oogenesis and fertilization, and the secondary oocyte is released when it is accepted or received by family, and it moves from infundibulum towards the ampulla, and it is surrounded by sperms. And one sperm got succeeded in making its penetration, its entry inside the cytoplasm of the nucleus by the process of acrosomal reaction, where the sperm had released a secretion or an enzyme hyaluronidase to dissolve the zona pallidus of this egg and make the penetration. It is had penetration inside the cytoplasm of the egg. So already here lies a haploid nucleus of egg, and when the sperm makes its successful penetration inside the cytoplasm of the egg, now there are two heads, there are two nuclei. One nucleus from secondary oocyte, that is egg, and other nucleus is from sperm, which is also haploid in nature. Now there occurs the fusion of two nuclei and a diploid zygote is formed, and these are two haploid in nature. When they fuse together, a single zygote, a single celled zygote is formed with a single nucleus, but this nucleus is now diploid in nature because it got half number from egg and half number from sperm nucleus. So now actually our topic starts from this stage, when now this diploid zygote have to undergo the repeated divisions in order to form a multicellular structure which is known as blastocyst. So this is cleavage. So first of all, after the fertilization, after the formation of zygote, within 2 days or after 30 hours, the first division, mitotic division occurs, and the single celled zygote gets divided and form two cells, and these two cells are known as blastomeres. Then it undergoes next mitotic division, and these two cells, they give rise to four cells. And after that, the Next division occurs within 40 hours of fertilization. The third division occurs about after 3 days, that is after 72 hours of fertilization. During these cleavage divisions, the young embryo moves slowly down towards the uterus. क्या होता है, अब ये फेलोपियन ट्यूब में होता है when it undergoes different stages of divisions. First within 2 days or after 30 days, second after 40 hours, and third after 72 hours or after 3 days. And with along with these divisions, it slowly moves towards the uterine wall or toward the uterus of the egg. So students, as you know that during these cleavage divisions, the young embryo moves slowly down towards the uterus. So now when this multicellular structure, which is known as blastula or blastocyst, it is also here, in other words, in zoological term, it is known as embryo. So what is embryo? Embryo is the developing fetus, but at its early embryonic stage. Embryo is the developing fetus. It's also a developing, it is a fetus but developing fetus, but at its early stage of development. At its early stage of development. At the end of fourth day, that is at 96 hours, the embryo reaches the uterus. You should understand, you should memorize it, you should know it, that 4 दिन के बाद, that is after 96 hours, this reaches to the uterus of the mother. At this stage, it have either 8 or 16 blastomeres or cells, and this solid mass of cells, it is now known as morula. Here, when it reaches to the uterus of the mother after 96 hours, it is in the shape of a mulberry, hence it got its name as morula, because it resembles with a little mulberry. That is why it have got its name as marula. And here at this stage, it has at either 8 cells or 16 cells. So now this marula at the stage of 8 cells or 16 cells, it now reaches to the uterus of the mother, and at this stage it is known as marula, and this all happen within 96 hours or after 4 days. So here it is another a pictorial graph by which I will try to understand you what actually happened. So firstly, I have showed you that when sperm and egg in the ampulla of this fallopian tube, they fuse together and a diploid zygote is formed. So this is the diploid nucleus, and as you know, there on the polar bodies are also formed during the oogenesis, and these are those polar bodies which lie in the perivitelline space, and this is nucleus, and this is the outer covering of this egg zygote, which is known as zona pellucida. So in case of zygote, the zona pellucida, it just acts as a protective covering for the developing this nucleus, for the developing zygote. So this is the first division, and two these blastomeres are formed, and one polar body got degenerated or dissolved. Now only one polar body remains here, and this is the zona pellucida, the outer covering of developing this zygote, which is protective in nature. And this is the first cleavage division, which happen after 2 days or after 30 hours. And then these two blastomeres, they undergo next mitotic division, giving rise to 4 cells. These are the 4 cells, and here it is zona pellucida still lying outside for protective nature, for protective coverings. And then these 4 cells, they undergo a next mitotic division, thus giving rise to 8 cell stage, and here it is the zona pellucida covering. And then these 8 undergo next division, thus forming 16 cells. 16 cell structure, and this structure resembles with a little mulberry type, hence it got its name as marula. And at this 16 cell stage it is known as marula, and this is the zona pellucida covering outside this marula, which is at the stage of 16 cells. Now this undergo again into the divisions, and their structure is formed near about of 64 number of cells. And at this stage where which resembles like this structure, where a mass of cells or this blastocyst is formed, a structure is formed which is having a number of cells equivalent to 64, and this type of structure is now known as blastocyst or blastula. So actually we have to read the development of embryonic development up to this stage, that is blastula stage and blastocyst. So students, here we will know a little bit about what blastocyst or blastula is, how like it is, how is its structure. We should know a little bit about its structure. So blastocyst formation, as you know, students, this is the mass of cells lying at the one end, and this is the other end of this blastula, and here inside it there is a blank space which is known as blastocyst cavity or blastocele, and there is a single line of cells which are known as trophoblasts, which are known as trophoblasts. So these trophoblasts are there, and this is the inner mass of cells, and this is a cavity which is known as blastocele or blastocyst. So here we have to know in this blastula or blastocyst that the site which possess this inner mass of cells along these trophoblasts, along this single layer of trophoblasts, this is known as animal pole or embryonic pole. So why it is known as animal pole or embryonic pole? Because this inner mass of cells which are lying at this side, at this end, they are now helping in the development of, and they are now helping in further embryonic development. When these cells enter further embryonic development for in order to give rise to full term forts, that is why this side is known as animal pole or embryonic pole. And to the opposite of this pole, where this mass of cells is lacking and only these trophoblasts, single layer of trophoblasts is present, this one and this side is known as abembryonic pole or this vegetal pole. And mostly and now what are about these trophoblasts? These trophoblast cells, they provide nutrition to the developing this blastocysts or developing forts. So this was a little bit about this structure of blastocyst. So students, you have to simply memorize that there is a single lining of cells around this blastocyst which are known as trophoblasts, and to the inner of it at one side there lies an inner mass of cells which help in further embryonic development. Thus this side is known as animal pole or embryonic pole, and to the opposite of it there is another pole which is known as vegetal pole or abembryonic pole, this side. And in the center of this blastocyst or blastocele blastula, there lies a cavity space which is known as blastocele or blastocyst cavity. Actually it lies filled with a fluid which help in the transmission of cells. And finally these trophoblasts, they are here for providing the nutrition to the developing these foods. And one more thing, these trophoblasts, they also at the one end they are providing nutrition to the developing embryo, and at the same time they also help in the formation of placenta, in the formation of chorion and embryon. And what is chorion, embryon, and placenta? We will read about these things in the coming class. But here I may tell you that it is simply a junction, a bridge, a connection between developing foods and the mother, where from the developing foods get its materials, its this food, etc., from the mother. So this trophoblast cell lining, they are very much important. Besides they are providing nutrition to the developing foods, they also help in the formation of amnion, chorion, and placenta. And students, these cells, which these trophoblast cells, which here lie with the touch with the connection of the innermost of cells, they are known as cells of Rober. So cells of Rober, these are actually the trophoblast cells which lie in contact with these innermost of cells. They are known as cells of Rober. And this is here, this is zona pellucida. Now it starts rupturing, it starts breaking. Up to this stage, the zona pellucida was in close contact, but at blastocyst stage, it start rupturing, it start breaking, because this blastocyst or blastula now had to embed into the uterine wall, and the placental formation, amniotic and chorionic formation have not start. So for the reason this zona pellucida now starts breaking. So students, this was all about embryonic development during the development of foods inside the mother swarm, inside the womb of the mother. So that was the embryonic development after blastocysts formation, blastocyst stage. Hope you all have to understand this. You would have understand this lesson. And if there are any queries, do call me or message me. Thank you.